Assalamualaikum warahmatullahi wabarakatuh. In this video, we will present about the Gcirms instrument. This is our group members, Siti Khadijah binti Nurhaid, Nur Hazana binti Kamarul Hisham, and Balkis Nur Akila binti Muhammad.
Gas chromatography isotope ratio mass spectrometry (Gcirms) is a specialized application of isotope ratio analysis, in which mass spectrometric methods are used to measure the relative abundance of isotopes in a given sample. The isotope ratio mass spectrometer (IRMS) allows the precise measurement of mixture of naturally occurring isotopes. Most instruments used for precise determination of isotope ratios are of the magnetic sector type. For the example, magnetic sector mass spectrometer used in an isotope ratio analysis through thermal ionization.
GC is a separation technique where a mixture of compound is vaporized and injected into a column that separates the components based on their interaction with the stationary phase. IRMS is a technique that measures the relative abundance of stable isotope in a sample. In Gcirms, the process begins with gas chromatography, where the sample is vaporized and injected into a column. The compounds in the sample are separated based on their interaction with the column and carrier gas. As the compound exit the column, they enter the mass spectrometer where they are ionized and separated based on their mass to charge ratio. The detector then measures the isotopic ratio of the compound. This is the whole diagram for the Gcirms instrument.
From the diagram, there is four main components of Gcirms. The first component of Gcirms is the gas chromatograph. It includes an injector, a column, and an oven. The injector introduces the sample into the system where it is then separated into individual compounds in the column. The oven controls the temperature to ensure optimal separation. Next, the separated compounds enter the combustion or pyrolysis interface. Here, the compounds are converted into simple gases such as CO2 and 2OH2, which are suitable for isotopic analysis. The gases then move to the isotope ratio mass spectrometer. The IRMS has an ion source that ionizes the gases, a magnetic sector that separates the ions based on their mass to charge ratios, and Faraday cups that detect the ion and measure their intensities to determine isotopic ratios. Finally, the data system analyzes the isotopic ratios detected by the IRMS. It processes the data and provides the final results, which are used for various analyses.
For the application, in 2000, IRMS was used to assist in the investigation of an unidentified set of human remains that were found by the Great Salt Lake in Utah. By looking for differences in the oxygen isotope ratios in hair segments, it was possible to predict where the individual had traveled based on changes in her drinking water isotopes. This prediction ruled out some potential locations and highlighted others for additional investigation. The analysis eventually led to the discovery of a missing persons report that fit many features of the remains, and a DNA test between living relatives confirmed the identity of the unknown remains.
The next application is to provide a means of obtaining a more in-depth understanding with respect to the traditional analysis of bulk products. Examples of application are reported for a single amino acids in wheat and durum wheat samples to discriminate between organics and conventional agricultural practice, vanillin to discriminate between natural and synthetic samples, fatty acid to identify the adulteration of high-value oils with cheaper oils. And lastly, wine volatile compounds to reassess the water status in vineyard. In relation to a wine, the Gcirms method was used to analyze 13 carbon or 12 carbon of high alcohols, for example, glycerol. From the correlation of delta 13 carbon of high alcohol compounds such as 2 or 3 methyl butan 1-ol with that of 1 ethanol R squared equal to 0.829, they were able to improve the detection of chaptalization.
Lastly, adulteration detection using Gcirms involve analyzing the isotopic ratios of key components in products like honey, olive oil, wine, and fruit juice to identify the presence of non-natural additive. In honey, for instance, Gcirms measure the carbon isotopic ratios of sugar. Natural honey has a consistent isotopic signature reflecting its botanical origin, whereas adulterated honey mixed with cheaper sugars like cane sugar or corn syrup shows different isotopic values. This method is highly sensitive and precise, capable of detecting even small amounts of adulterants. By comparing the isotopic data of the product to known standards for pure and adulterated substance, Gcirms can confirm the authenticity of the products. This ensures that consumers receive genuine products and helps maintain the integrity and trustworthiness of food products in the market.
That's all from us. Thank you.